All right, thank you for the invitation. And I admit when I received the invitation, I wrote back and I said I greatly appreciate it, but you clearly made a mistake because I use my iPhone I need to make, and they said no, we want people with different perspectives, so please welcome. So I will mention some of the modeling that we've done, but I haven't done any of it. I'm a pathologist by training. And I'll just take you through some of the work that we have done, that we are doing, and I'll end with where we are going. So this, to me, is always been my interest. I have a parent who died of metastatic colon cancer when I was a kid, and I was always interested in how and why. So the how, of course, could be mechanistic, many things that way, a lot of very interesting science by many people in this room and others. I think the why gets a little more into the modeling, could even be philosophic. Why does this happen? A patient always says, why did this happen? I didn't smoke, why did this happen to me? But the how and the why is what has always driven me. So let me give you a little historical perspective. Autopsies used to be the only way to diagnose disease. Somebody would have signs and symptoms and doctors would care for them, but ultimately when the patient died, it was in a theater where the autopsy would be done, and all the students and physicians would be surrounding, and they would look at the organs and they would learn why the patient died. So it used to be a central way to understand disease. Now, modern medical science has really replaced the need for autopsies, because now you can figure out many of these things while the person is alive and make an intervention. And that's a good thing. Of course, the ability to, the solving of the human genome and what that has meant for understanding cancer, I think, cannot be understated or overstated. And of course, once you can sequence the genome, you can sequence cancer genomes. And that's really given us such a view of the patterns of cancer, what genes are affected, how they're affected in different tumors, what are those patterns, so genome landscapes. But with all of that data, moving away from Sanger and getting into next-gen methods and start looking at different samples in the same person, it's really made autopsies come to the forefront again. So it's an ideal way for, remember, my goal was always to study metastasis, and when I was starting my training, a lot of metastasis samples just weren't available in the archives in pathology. So I just took advantage of the fact that I'm a pathologist. I knew how to do an autopsy. I'm a doctor. I know how to speak to a patient. And I had done a postdoc, and I was interested in genes, so I thought I'd just put those things together, and I started this autopsy program in 2003. I've been doing it ever since. And it's really an ideal way, in my mind, to study metastasis in humans. And also why treatments fail. Those two things are interrelated to each other. If treatments didn't fail, if tumors didn't metastasize and they could be controlled to where they are, then we wouldn't have such a mortality rate like we do. Research biopsies, if you want to obtain a biopsy of metastasis for research, that could be unsafe for the patient. The lesion could be located someplace that it's just not safe to put a needle. Intratumoral heterogeneity, we're hearing about that a lot, and I will talk about that as well. This topic can be addressed by an autopsy approach, because you don't have one sample, you have everything. You have enough tissue to support multiple omic studies. So this is something that came up at Memorial. Christina Leslie was leading a discussion about, little microscopic samples that have been used for targeted sequencing at Memorial, and how do we use those samples for everything else we want to do. And everybody would have to make their case why their assay is the best one. Here, there's an embarrassment of riches. You have so much tissue, everybody could have some, there's still some left over. It reduces the need to infer what is going on in a sample, because you have the sample, you have everything. I think Dan Landau made a point this morning, I think it was a really good one, that there's always a place for statistical inference and functional assays and observation, and I completely agree with that. This helps because it gives more data to build those statistical models on. So having more data that you can observe and characterize and measure can only be a good thing. So what do? It's not rocket science. Basically, you take everything that you see, and usually we found the fatigue factor is about 2 hours. That's when everybody gets tired and they just want to stop putting tissue in tubes. So cases usually take about 2 hours, and we take all of the tumor we see. If the primary is there, we take the entire thing. The metastases, we take as many as we can collect, or at least what is representative if the disease burden is huge. Samples of all normal tissues. We get biofluids. You can get blood, high-quality blood post-mortem. We get ascites, urine, CSF, everything. It's all high quality, circulating tumor cells. And these cells are still viable. That's one thing to remember. People always think, well, the person's dead, therefore it's all dead. That's not true. The individual is dead, but their cells are not necessarily dead. And we've made xenografts and cell lines from many a case and many different tumor types. I think the most extreme example is a case that was on 72 hours, 3 days post-mortem. And the reason this happened was I was not at the case, I was traveling, and so my lab, just knowing what I always do, they submitted a sample and they all grew as xenografts. So it just shows you never know. So what do? We don't, again, I don't want to ever not take something, because that could be the magic sample that has the magic finding. So we do multi-region sampling, and we keep everything, and we sample everything in a way that we can always reconstruct the 3D characteristics, the topology of the tumor and the mass. This happens to be a metastasis of a woman with breast cancer for all different tumor types, primary and met. So they're sectioned into 1 cm thick sections. There are the sections there that you can see, and then we'll take one of those that's hatched in the box, and that's cut into little squares or semi-rectangles, and then that is bisected, and you have a section that goes, that's formalin fixed, and you have a section that is snap frozen. So if you take what's in the tube and sequence it, you have a histologic correlate. If you're looking under the microscope and you see something interesting, a morphologic interesting pattern, you can go to the tube and sequence what you have. So it's an incredible way to maintain all that information. So I have used this approach to study pancreatic cancer, and I did not intend to study pancreatic cancer when I started my career. I wanted to study colon cancer because of my father, but it actually turns out it wasn't an easy tumor type to work with for this approach, because patients would have a colon cancer, and that tumor would be removed, and then they might have a recurrence 3 years later in their liver, and then they would come to Hopkins, where I was at the time, and have a partial hepatectomy, and it was just too complicated. Whereas pancreas cancer, not only was Hopkins a center of pancreatic cancer research, one of the original, patients with pancreatic cancer, just because of the natural history of the disease, it's very unfortunate for patients, but it makes it very amenable to studying metastasis. As metastasis is a common feature in the natural history of the disease. 80–90% of people will develop metastasis. 50% of patients present with metastasis. Many patients, as I said, they present with metastasis. They still have their primary carcinoma in situ when they pass away, so you have the primary to compare to those metastases. So that's important. And many features of pancreas cancer are applicable to other solid tumor types in terms of natural history, carcinogenesis, stepwise progression models, whatever. So while I'm going to make the point later on, don't compare tumor types, at the same time, there are some things that you can compare among tumor types when it comes to very fundamental questions, I think. So pancreatic cancer is a multifactorial disease. Compared to other tumor types, as you see, I will just walk up here. This is cancer, and this stuff in the middle are inflammatory cells and stroma. And pancreatic cancers make a very exuberant stromal response, and the stromal response itself is responsible for driving a lot of the phenotypic properties of a pancreatic cancer. And of course, of all the things that can cause pancreatic cancer, they can be broken down into cell extrinsic and cell intrinsic. I study genetics because that's what I'm interested in. Without a mutation, you don't have all the other things that follow. Genetics to me are the bedrock of what starts it. and then I think everything else takes perhaps precedence and takes over later on. And so I study the genetic part, and genetics are nice because it's kind of a no-lose situation. If there's a mutation, that's great, but if there's not a mutation, it still provides a way to lineage trace within your tumor. So now we'll get a little bit into what do we think about this. So there is heterogeneity, and I think even in this session, heterogeneity of reported definitions of heterogeneity. What is the definition of heterogeneity? And depending on what you define it as, you can get a very different result. So there is expected variation in a tissue or a tumor, and then there could be heterogeneity. So heterogeneity, that could be within the patient, across patients. It could be the tissue type of origin. It could be the cell type of origin. It could be genetic heterogeneity, epigenetic, transcriptomic, neoepitopes. You see, there's many ways of heterogeneity. Within genetics specifically, are we talking about driver genes? Are we talking about passenger genes? Any mutation, driver genes only, et cetera? What are the thresholds for calling a driver gene? And copy number alterations, what is the definition of what is a true copy number alteration versus what is just aneuploidy and noise? So I also try to think out, so I'm giving you my definitions before I show you my data, okay? So again, the last speaker nicely pointed out there's different kinds of heterogeneity, and that's a very valid point that always needs to be driven home. There's, of course, within a single patient, there's heterogeneity between patients. That's the reason why there are different responses of patients to the same therapy. You know, they could have genetic differences or innate differences. And then are there differences in driver genes specifically versus in any mutation that's present or absent? In my lab, we focus on driver genes. Now, driver genes alone could have an entire symposium on it. How do you define a driver gene? Well, I'm not in the business of defining a driver gene. So what we've done in our lab is basically to constantly curate the literature for what are cancer genes that other people are reporting based on statistical data, whatever is a cancer gene. So we have a list of about 6,000 genes in 598 pancreatic cancers that have been either whole exome or whole genome sequenced through a variety of studies. We've accounted for redundancy of samples in different studies, et cetera. This list is, I think, even a little outdated now that I'm looking at it, but as you can imagine, there aren't 6,000 driver genes. So clearly our list is very broad and probably has a lot of noise in it. So, but we're increasing the chance that we have real driver genes, and then we have perhaps the tail, rare driver genes, and then there could be some things that are just inherent noise. But that is our list that we're working on. And then how do we think about heterogeneity in a single patient? We think of it different ways because each one for pancreas cancer has different consequences. Okay, so heterogeneity within a primary tumor. So I'm talking about heterogeneity for driver gene mutations in a primary tumor. So that is the example indicated here by the different colored cells. You can have heterogeneity for driver gene mutations within a metastasis specifically. So in the first instance, that might matter for somebody who presents with an isolated primary tumor. In the second scenario, that matters to somebody who presents with recurrent disease and has a metastasis. That would be the issue. And then the third scenario is heterogeneity of metastasis-initiating cells in a primary tumor. So you can see you could have scenario one, where you have subclonal heterogeneity within a primary. That doesn't mean you have scenario three, because your metastases could be monophyletic, right? They come from one subclone. So this is how we try to think of the disease and categorize in a way that makes sense. Now, with that in mind, our question was, what is the extent that cells that see distant metastases are clonally heterogeneous at the genetic level, okay? And that question is important for pancreas cancer because, as I said, 50% of patients present with stage 4 disease. They're usually given gemcitabine, which in the classic study that resulted in FDA approval of gemcitabine, it resulted in a 3-week extension of life. So that's something, it's statistically significant in the study, but 3 weeks of poor quality life is not an advance. These patients are given gemcitabine or they're referred to hospice, and then they die, and the survival is between 3 and 6 months. So these people have nothing. So we need to find something for them, and that's what our question is. So because I've been doing autopsies a long time, and I've always been open to anybody, any scenario, never excluded anybody for anything, if you wait long enough, you get patients that happen to have the scenario where they presented and they were diagnosed and they either died too quickly or they denied treatment and they died, so they were never treated. So starting from patient PAM-01 to PAM-04, that's about 11 years of time to accumulate these 4 people. Just to give you an idea, like this is, you know, oh, let's not, you know, I didn't say let's do a project and here's 4 people that came last month. You know, these are hard to come by. So these are 4 patients, untreated, treatment naive, stage 4. So this is important because it gives us the opportunity to look at the natural history of pancreatic cancer without any exogenous bottleneck. So everything that's happening is what happened in the person, and it was no influence. So you can see the patterns of metastasis reflect the patterns of metastasis you see in pancreas cancer is usually the liver, lungs, and peritoneum. And for each of these samples, we extracted DNA and we did whole genome sequencing. And so we found, the short story is that we found no heterogeneity for driver genes among any sample in these people. So remember, driver gene, I'm talking about that 6,000 gene list. So that's a very broad net. We didn't find any heterogeneity for any of those. The trunks were long. You see the driver genes that are there. They were short, stubby branches. We validated by immunohistochemistry for protein products, copy number plots. You can see essentially there's red stripes, there's blue stripes. Now, somebody might say, well, what about this area here? That looks different. It is different, absolutely. That region did not contain any of those 6,000 driver genes. So that just says, again, the specific question was, is there heterogeneity for driver genes? Our answer is no. But that doesn't rule out, I mean, very interesting things I heard this morning about long non-coding RNAs and things like that. I think there's a lot of room for that there. And again, we're talking about pancreas cancer here. Now, we did find heterogeneity, if you use it in the sense of any difference. We did find heterogeneity between different samples. So this is one person where it's a Jaccard index. You're just looking at the fraction shared over all mutations between the 2 of them. And you see the numbers are very high, with a median index of 0.97 of all mutations that are shared among everybody. Now, are these mutations important? I don't know. Bioinformatically, we've run it through a number of algorithms. They all appear to be passengers. None of them were occurring anywhere that would have predicted to affect protein function, anything like that. They don't appear to create a neoepitope of any significance. And you can almost argue that probably makes sense because these people presented end stage, so the immune system likely failed at that point. So these are just passenger mutations, but that does count as heterogeneity if you just use the definition of if anything is different. So our next question was, and this gets to the quantification part finally, that why I'm in this session. Our question was, is this 0.97, if you looked at normal tissues or some control, would it be 0.99? Or would it be below that? So there is no ability to sequence the genomes of individual cells yet. So that's why we referred to modeling. All modeling was done in Martin Nowak's lab by Johannes Reiter and Ivana Bozic. And essentially they looked at what would be the expected homogeneity index or Jaccard index, I guess, if you looked at 2 different normal cells in the same organ, accounting for how many doublings it would have to go through, mutation rates, size of the organ, everything like that. And scenario 1 is neural tissue. because that undergoes a scenario where you have cells that stem cells develop during organ development, and then they terminally differentiate, and you have a brain. And that index is 0.03. Scenario 2 is when your self-renewing stem cells are compartmentalized, so like the intestine. And that index is 0.04. And then scenario 3 is hematopoietic tissue, where all of your terminally differentiated cells are all mixed up and flowing around the body. And that upper bounded at 0.2. So based on math, that is what we came up with. Now, right as we were had this paper in revision, Hans Cleaver's group, along with Mike Stratton, published a paper where they isolated single cells from normal tissue and created organoids and sequenced them, and they came up with the exact same numbers as the model predicted. So any two normal cells in a tissue are much more dissimilar than any two cells in a metastasis, okay? It's stated the other way, metastases are very homogeneous compared to normal tissue estimates. And this is metastasis-initiating cells. So remember I said we sample our tumors in a way that we can always go back in 3 dimensions and know anywhere in that tumor where that sample came from. And we also have the ability to look under the microscope. What do these different subclones look like? So notice again, see the numbers on the branches are not big. It's +1, -5. There's very few mutations that differ among these branches. And that goes along with that. There just wasn't much heterogeneity at all. But this patient, this is what sample PT1 look like. So PT1 is not the parental clone because the parental clone no longer exists, but it's closest in relationship to what that parental clone would be. And this looks like a prototypical pancreatic cancer. You have what's circled are the tubular glands of the neoplasm. There's a lot of stroma and inflammation. And then this is another sample of the primary tumor located 2 centimeters away. Looks completely different. You don't have to be a pathologist, I don't think, to know that the bottom looks different from the top. It's darker, it seems to be more crowded, whatever. So this is phenotype switching, and to have such a dramatic change in phenotype without any change in genotype smells very strongly of something epigenetic. As this was going on, the sequencing project, I had a GI Path fellow named Oliver McDonald, who's now faculty at Vanderbilt, and he's a very smart guy. So I said to Oliver, who was interested in epigenetics, why don't you follow this up? And this resulted in us having, nicely, they started as 2 separate papers, but it ended up as 2 separate projects started 5 years ago, but they ended up in the same issue of Nature Genetics as back-to-back articles because it just all sort of converged together. So this is PAM04. You see the tree is pretty boring, the long trunk and the little bushy trees. And we have the samples of the sample that was closest to that parental clone, the founder. We have the sample corresponding to the subclone in the primary tumor that seeded the metastasis, that peritoneal metastasis. So we can literally look at those. And by H3K9 methylation, there was no difference, whereas here is PAM02, which happens to be the person I just showed you on the previous slide with the histologic images of the phenotype switching. This is this patient, and you see very differently, the founder has methylation. The intratumoral subclone that gave rise to this metastasis has the beginning of individual cells that are losing methylation, and then the liver metastases completely lose methylation. This is a really robust finding. So how do we put this in context for pancreas cancer? If a patient presents with stage 4 pancreatic cancer and is found to have an actionable genetic alteration, it is worth pursuing because it's probably truncal. It's probably in all cancer cells. And here's an example to highlight that. This is a patient who was found to have a somatic BRCA1 mutation, but it was at a very high allele frequency, likely clonal, treated with cisplatin, and everything melted away. And this person is still alive 3 years later. So this is somebody that would have been referred to hospice, and now they're alive 3 years later with a good quality of life. So we want to do, our goal is to understand the disease so we can make better, more informed decisions. So context matters. Not only, of course, what is the tissue type you're thinking of. I think actually listening to our previous speaker's talk, I got some ideas about something, just the striking differences between lung and pancreas. You know, they're very different organs. Lung is nutrient rich, it's oxygen rich, and you have a tumor that's growing. You know, what are the selective pressures in a lung? In the pancreas, it's nutrient poor, it's microvascular, there's a very high interstitial fluid pressure. I mean, there's so many biophysical differences. And it just makes me wonder, is that driving these differences that you see in these two different tissues? So, but to get back to pancreas, some patients, about 10–15%, will present with a tumor that is small enough to be resected. The problem with those people is that about 90% of those will recur. You see, it's just a very horrible, problematic disease. So if the person has a resection, in the OR, the remaining pancreas is anastomosed to the bowel, and then the person is treated with adjuvant therapy, but then unfortunately most of them will recur. And they recur in the pancreatic remnants and or in distant sites. So again, going to our autopsy series, we found 10 patients where we have the original, the recurrence, and the metastases, and we did whole exome on all of those. So remember, I had just said that stage 4 pancreatic cancer is completely homogeneous. This is what happens when you have resectable pancreas cancer that's treated. It's completely, it's very heterogeneous. For driver genes, unequivocal driver genes that have robust functional and bioinformatic evidence that they play a role. Many of these have been validated by immunohistochemistry in the exact same tissue. So something's called a deletion. It's truly missing in the tissue. And I should actually say many things that were called deletions or altered by copy number changes did not validate by copy number alteration. So either the gene we looked at was not the target of that copy number event, or it's just noise. But I think it's a striking example that you have to look at the context, and how I think of this, perhaps my working model is, you know, the stage 4 patients, those tumors have undergone a clonal sweep, a clonal purifying sweep. And so it's a very homogeneous neoplasm. These patients, those tumors are more heterogeneous, and you're going to see the heterogeneity and the bottlenecks and whatnot. So this is a manuscript now. We have a lot of other data go along with it, but that's where we are. So what are our future directions? My future direction is to go boldly into all directions, okay? Just aimlessly going. Don't letting my misunderstanding keep me from doing anything. It worked for me with the autopsy program. I thought I would get 5 cases and get a nice paper and be done, and I never thought it would go the way it is. At Memorial, we are now doing this approach for every tumor type. It's not just pancreatic, because I think, you know, we're at the point now, myself included, anybody who knows how to use a computer can upload a list of genes and make a phylogenetic tree. You know, we need to move beyond that and start looking at fundamental questions that how do trees compare across different tumor types in comparison to a targeted versus an immunologic versus a cytotoxic therapy. How does the tissue of origin affect those patterns, things like that. We need to think bigger. So we are well on our way to answering that question, because we've done many cases so far, and for something else, a report I had to do, we have 8,000 specimens banked already. We are moving into experimental evolution because, again, we're inferring a lot of things that are happening, but there's some things you need to measure in living cells. Again, the concept of if you have a resistant clone, but that resistant clone and that mutation is in a cancer cell fraction of 10%, you know, what is going on there? And this allows us to address those questions of clonal cooperation. Is one cell with the mutation, is it sort of cooperating and driving, you know, 10 more cells around it that don't have the mutation? We can start to answer those kinds of questions. We can address the mechanisms, not how does resistance occur, you know, oh, they developed this mutation and now they're resistant to therapy. Literally, how does that mutation occur? We know sometimes they're pre-existent, There might be instances where they're not all pre-existent. How does that occur? Literally the mechanism of how the resistance occurs, and that includes the epigenetic, the phenotype switching. So this is actually a long-term evolution system. So we have cells growing in it now. You don't touch it at all. It goes on its own. The system maintains every possible feature. So we have cells in there more than 100 days growing. They're viable. You're seeing peaks and valleys, that there's just turnover and dynamics going on. And every 2 days we take a sample. So we have the ancestral and a fossil record. And that's the direction we're going to move into. Who knows evolution better than the people at the Museum of Natural History? So I just went to the head of paleontology and said, hi, and this is who I am, and they were so welcoming. So we actually have a couple of collaborations ongoing, really exciting. One of them is looking at comparative genome sequencing, because I'm very interested in how has the cancer phenotype evolved across species, across the tree of life. Another fundamental question that we're trying to move into. We're finishing up our first study comparing primate to human with some really interesting results. And I have to say, you know, they don't do cancer, but they understand evolution. If I showed them something complicated, they say, oh, it's clearly an example of that. So-and-so published it in 1970, whatever. And they end up being, you know, 6 months later and a lot of money, they end up being right. We have, Raul saw this, I proudly show him, we just started a Hydra colony in my lab. Because Hydra gets, so you're saying, what the hell is Hydra and where is it? Here's the Hydra. So it's, you know, it's like a little polyp and it's got tentacles on its head and it's making a little bud. It asexually reproduces, but it's a metazoan organism and it gets tumors. So it's the simplest known complex organism or multicellular organism to get tumors. So humans are so complicated, let's break it down into something more simple. So I said, again, let's grow Hydra. So it cost me $9 in a wine cooler on Amazon Prime, and we're up and running, and they're growing and reproducing happily. So that's where we're going. That's where we've been. You know, everything I'll probably be publishing in the next year or so represents data from cases I've accumulated over the past 10 years. But moving forward, a goal is to go big, you know, go big or go home. Ask the big questions, the fundamental questions, questions where modeling and math are required to really make a difference. So here's my amazing lab and my acknowledgements and support. I think Martin Nowak's getting a lot of credit in this conference, and that's it. I'm happy for any questions. I kept on the time.